United States Department of Agriculture.
I want to thank everybody for joining the session today and the 3R's Symposium organizers for their kind invitation to be a part of this wonderful event and serve with our austere and outstanding PIs and scientists and presenters today. So I have a basic plan in mind that I'd like to pursue in the next several minutes, and I want to begin with showing you some of the fundamentals of how light affects circadian regulation of neuroendocrine and metabolic and physiologic parameters of animal health and well-being. So I want to start by showing you some of the fundamentals of how light affects circadian regulation of neuroendocrine, metabolic, and physiologic parameters of animal health and well-being. And this will include studies on research animals and humans. And from there, we'll segue to what we currently understand about light cycle perturbations in the context of experimental changes in lighting and reverse adverse impacts on research animal health and well-being. And then I'd like to emphasize some practical approaches on how these things relate to animal welfare, as well as rigor, reproducibility, and translational potential of studies. Finally, we'll end up with a discussion on how to improve animal welfare and rigor and research that require experimental lighting changes. So I'm going to get at this, and I'd like to turn off my ugly mugshot for a moment. I'm stopping my video, but can you still see the presentation, everyone? So by way of introduction, then, all humans and most animal species evolve under a major geophysical event, and that is the rising and the setting of the sun. And for thousands upon thousands of generations, our bodies are exposed to the presence and absence of light on a daily basis. And also the waxing and the waning through the seasons. So we've developed an internal way to respond to light and darkness, which is really profound in principle, and it impacts the nervous and endocrine systems throughout a whole 24-hour day, as you see here in this slide. In fact, based on our current knowledge, we know that light and light-dark cycles regulate in major ways just about every biological rhythm in our mammalian systems, from when we give birth to when we pass, and about everything in between is shown here. So there are four basic types of biological rhythms: circannual rhythms, which have a cycle of about a year, like migrations and hibernations; infradian rhythms, with a cycle less than a year, but longer than a day, like the female menstrual cycle; ultradian rhythms, these also have a cycle less than a day, like eating cycles. But arguably the most profound of all the biological rhythms that influence life on our planet are circadian rhythms, which have a cycle of about 24 hours per day, as we both know, all of us. And these include sleep-wake cycle, body temperature cycles, and the melatonin rhythm that I'll discuss in a few minutes, as well as many of the rhythms shown on this slide. So in the early 1980s, the work of George Brainerd and Russ Ryder at the UTHS San Antonio was some of the most important to emerge. And this work laid the groundwork for lighting protocols that we include in our American Guide for the Care and Use of Laboratory Animals. Their findings are sometimes referred to as a holy trinity of light. That is, light influences circadian rhythms of physiology and behavior in an intensity, duration, and wavelength dependent manner. So briefly, light enters the eye and it passes through the lens to stimulate the retina. And photic signals are transmitted via the primary optic tract to the thalamus and then onto the visual cortex, allowing us to see the world and to do our work. In contrast, there is a separate pathway that extends from the retina and the optic chiasm to a non-visual part of the brain in the anterior basal portion of the hypothalamus. It's called the suprachiasmatic nucleus, or SCN, or master biologic clock. And it's shown here in this slide in blue. This paired nuclear group is located above the optic chiasm and near the supraoptic recess of the third ventricular recess. allowing it to readily receive photic signals from our retinas. There's a short projection from the SCN to the periventricular nucleus, and then a long descending multisynaptic projection all the way down to the upper thoracic region of the spinal cord. The retinohypothalamic tract pathway, or RHT, then leaves the central nervous system through the superior cervical ganglion, and postganglionic nerve fibers climb up the vasculature to innervate the pineal gland, which is located in the epithalamus. And it's shown here in the green area. Well, the pineal gland sensitizes and secretes a variety of compounds, but really the most thoroughly studied is the circadian nighttime neurohormone, the 5-methoxyindole, otherwise known as melatonin. And this figure on the bottom right shows the systemic melatonin levels that are high at night and low during the daytime. They're also low when animals are exposed to light at night, or what we sometimes refer to as LAN. This light-dark dependent entrainment of the SCN, the master biologic clock, regulates circadian rhythms of metabolism and physiology in all mammals and many other animals for that matter, and it's associated very, very closely with animal health and well-being. All of this functions in a relatively straightforward manner, if you can believe it, to provide two forms of information broadly through our nervous system. That is our sense of vision and biologic time. So this is the photoreceptor system shown here on the right, where the rods and cones adjacent to the plexiform layers synapse with amicron cells, and then the retinal ganglion cells. The majority of ganglion cells are doing the work of the visual system. However, an occasional ganglion cell contains the photopigment melanopsin, indicated here in blue, discovered in 1996 by Dr. Ignacio Provenzio and his team at the University of Virginia. And these cells are called the intrinsically photosensitive retinal ganglion cells, or iPRGCs, and they comprise only a small fraction of the total ganglion cell population, actually less than 5%, depending on the species and the method of estimation. iPRGCs are capable of detecting light and darkness completely independent of the rods and cones. And this is a very, very important point. The iPRGC melanopsin cells have their own pattern of wavelength sensitivity or action spectrum. It's like a fingerprint, if you will, and there are at least 5 subtypes known to date. And again, the concentration and subtype is species dependent. And in 2002, Dave Burson and Samar Hatter conclusively demonstrated the existence of these cells in mice. And more recently, in 2014, Dave Whitmore's lab at University College in the UK demonstrated the same setup, types of cells in zebrafish, interestingly enough. So the discovery of the melanopsin iPRGC system is a crowning achievement and has led to a revolution in our understanding of circadian photoreception, and is beginning to guide scientists into new uses of light in general, but specifically today, blue-enriched LED light, which is the most common type of light technology that we're changing to in the community, home, workplace, and the vivarium setting. And these papers shown here in the lower right of the slide discuss some of the seminal findings. This next figure is something not from our laboratory, but rather from the Jefferson University Light Lab of Doctors Brainerd and Hannafin. And it represents the findings of over 10 major studies in both humans and other species, including non-human primates, rats, and mice. The collective data is shown here. So if you have a normal color vision, as most of us do, you can see the spectrum at the base of the slide. That's your 3-cone system working with a peak sensitivity of 555 nanometers. And this is known in the neural literature as the standard observer. We have the photopic system or daytime system that utilizes cones and are capable of color vision and are responsible for high spatial acuity. There are 3 types of cones, as many of us know, the short, medium, and long wavelength, or SMNL cones for short. In contrast, however, the scotopic or nighttime system employs rods primarily. They're responsible for vision at low light levels. And rods don't mediate color vision, and they have very, very low spatial acuity. So at dawn and dusk, when light levels are low and both rods and cones are jointly operational, this time of day and the system that's in play is called a mesopic system. In the field of photobiology, getting back to what I mentioned once before, the action spectrum is one of the most important principal tools for identifying how the chromophore melanopsin initiates a light-induced response. It was a Brainerd laboratory in the late 1990s utilizing action spectrum studies in animals and humans that hypothesized that this non-visual IPRG system is the principal entrainment system for circadian, neuroendocrine, and neurobehavioral regulation, to include neuronal gene expression. Now, for regulating the circadian system, the action peak or the action spectrum is this peak in the center shifted over to the left portion of the spectrum that appears blue to our visual system in the 446 to about 482 nanomolar range. Photobiologists have evolved a set of refined approaches for determining action spectrum that are applicable for all light responsive organisms. So now we come to kind of the crux of the matter, if you will, in terms of light cycle perturbations and experimental changes in lighting. Our initial studies, which began back in the late 80s, if you can believe it, showed that a dark phase light contamination in animal rooms of only 2 lux resulted in dramatic suppression of the circadian nighttime melatonin signal, which resulted in human and research rodent tumor growth rates that were 2 to 4 times faster. This work, beginning over, as I mentioned, over 30 years now, was the first experimental evidence in support of the now over 30 epidemiologic studies in humans showing that LAN, or light at night, causes an increase in breast cancer growth and development. As a result, in 2007, the World Health Organization classified LAN as a probable class 2 carcinogen. This has been more recently upgraded, by the way. So these studies included work in the area of LAN influence on animal circadian rhythm alterations in metabolism and physiology, signaling pathway changes, and clock gene expression. And the publications on the bottom right are some of the later work supported in full or part by the NIH, the Department of Energy, and our own ALS glass program here in America. So as I mentioned previously, light and light dark cycles impact just about every animal neurohormonal, neurobehavioral, metabolic, and physiologic factor in all life on the planet, as it's summed up in this slide. And these include the acute effects of light, like changes in melatonin secretion, dietary and water intake, body growth rate, shown on the lower left, as well as the longer term effects, like circadian phase shifting and entrainment concerns, shown on the lower right. Indeed, based on the currently available data, alterations in light and light dark cycles and light technologies significantly influence animal health and welfare, as well as the rigor, reproducibility, and the transitional nature of scientific investigations, that is, scientific outcomes. So I'd like to encourage the audience to access the following 3 what I feel to be outstanding articles that provide superb guidance regarding the impact of light and light cycle perturbations, to include experimental changes in lighting. And the first is the Rob Lucas and his team consensus article, which details current methods for measuring vivarium lighting and practical guidance for implementation in animal husbandry and experimentation, and quantitative guidance on the appropriate light exposure for laboratory animals. The guidance provided here has the potential to improve data quality and contribute to reduction refinement, helping to ensure more ethical research animal use. The second article by Larry Brown and his team provides an overview of the methods used to quantify circadian disruption across the literature as a result of light perturbations. It focuses primarily on the laboratory mouse. These methods include paradigm power, variability in light onset, light phase onset, activity bouts, and interdaily stability, intradaily amplitude, and relative amplitude of light. And finally, I really encourage folks to consider looking at Stu Pearson's review article, which provides a wonderful overview of the visual and non-visual systems of the laboratory mouse before discussing practical considerations for the use of light for researchers and vivarium staff. Importantly, all three articles provide a wealth of information, again, pertaining to light cycle perturbations in the context of experimental changes in lighting, and at the same time detailing methods to reduce adverse impacts on research animal welfare, as well as rigor, reproducibility, and translational potential of scientific investigations. So, in summary, for these first two parts of the talk, light in an intensity, wavelength, and duration dependent manner regulates circadian rhythms of neurohormonal and neurobehavior systems in all animals, including research animals, obviously, in the vivarium setting. This occurs via the visual system of the primary optic tract and the non-visual system of the IPRGC and retinohypothalamic tract. Secondly, light cycle perturbations to include experimental changes in lighting and lighting technologies, such as exposure to light at night and our current transition to LED solid state lighting technology, also impacts animal welfare. And finally, appropriate species-specific light and lighting protocols positively enhance animal health and well-being. Again, as well as the rigor, reproducibility, and the translational potential of our scientific outcomes. So, in stepping back a moment, let's look at how light and lighting technology has impacted both us as humans as well as our research animals, our heroes in the vivarium setting. And one of the most profound events in thousands of generations of life on our planet was the advent of the electric light bulb a mere 150 years ago. This substitution of artificial light for natural sunlight completely reversed mankind's exposure to light. Now, instead of spending nearly 90% of time out of doors exposed to natural blue and rich sunlight, we spend it inside, and 50% of the time spent outdoors is actually within motor vehicles. Can you believe this? Indeed, exposure to daytime sunlight enriched in the blue appearing portion of the visual spectrum is really minimal, as reported in the articles in the lower right-hand side of the slide. So with the previous points of this discussion in mind, I'm moving forward to the final portion of the talk, and that is, what are some of the practical approaches regarding vivarium lighting and lighting protocols as they relate to animal welfare? First, consider eliminating all animal facility light at night, as we discussed early on in this talk. As a reminder, the publications on the bottom right represent some of the later work supported in full or in part by the NIH, the Department of Energy, the ACLAM Foundation, and the ALASH Glass program. With the previous points here, I'd also like to ask you secondly to consider transitioning to LED solid state lighting technology. So actually it began in 1997. We were looking at LED light compared to fluorescent light at nighttime and its impact in the vivarium setting, the light at night exposure on animals. But in 2010, our team began to investigate the impact of LED lighting at daytime, more specifically in the blue enriched portion of the visible spectrum, lighting at daytime, which I sometimes have called in my publications BLAD, blue enriched LED light at daytime. Its impact not only on human cancer, but research animal metabolism and physiology in general as it relates to animal health and well-being. And BLAD lighting is the most commonly transitioned to lighting technology in the community, home, and workplace and vivariums today for obvious reasons. Cost is lower, higher efficiency, superior spectral control, the sturdiest size and weight of this solid state technology. There's literally no noise and vibration, especially with newer lights, and no flicker, and dramatically longer operating life. Listed below on the slide are many of ours and other publications pertaining to the lighting technology. So I mentioned a bit earlier that the most common LED light we're currently transitioning to globally is the blue enriched LED shown here in the upper right, whose spectral power distribution is shown here in the bottom right compared to the standard white fluorescent light shown on the left. Spectral power distribution, or SPD, is the amount of power in microwatts per unit area at a given wavelength of light. And what you see in this slide is that while both lamps are broad spectrum, the LED lamp shown on the right is significantly blue enriched compared to the fluorescent lamp on the left, if you will, which is markedly more warm at 2800 Kelvin compared to the cool LED on the right at 5000 Kelvin. Though the total luminous flux, or lumens, a measure of the total quantity of light, is actually a bit lower in the LED. Today, as we transition to this LED light technology, peak emission in the short wavelength light, the SWL range of the visible spectrum, is really somewhere around 450 to 470 nanometers, a little bit more sometimes, depending on the measurement scheme, to produce a very bluish light. And due to the nature of LEDs to be tunable or combine different phosphors at different intensities, this lighting technology now may provide colors, if you will, at the full range of the visible spectrum. Interestingly, nearly all the work regarding daytime exposure to LED lighting began with ALS and the glass-supported grants that we received. And if one may sum up all the work of the top 12 publications on this side, essentially, animals subjected to blue-enriched LED light at daytime had lower dietary water intake, slower growth rates, and neurohormonal and neurobehavior and physiologic indicators, all of a more youthful phenotype. And finally, let's keep it simple. Let's follow a proactive regimen, if you will. In these last few moments, I know I'm a little behind probably, but I'll continue on here. Let's be proactive. Lighting expert consensus work from 2011 today, 2024, mentioned earlier, determined that lower LED daytime vivarium light below levels of 300 lux skewed toward the blue spectrum were generally considered the best in terms of the maximum stimulation of the visual and non-visual systems for research animal health and well-being, and hence scientific outcomes. Conversely, exposure to as little as 0.1 lux of light at night was very bad. That said, most institutions have already taken steps in the direction of LED technology. We recommend regular monitoring and recording of light intensities, particularly in both the room as per our guide, but also within the microenvironment of the caging systems. The tools are readily available, including inexpensive spectrophotometers and a host of available room monitoring systems. Finally, it's a team effort. Don't be afraid to consult the experts in the field. The information that's been provided here has addressed light cycle perturbations in the context of experimental changes, practical approaches, and how they relate to rigor, reproducibility, and the translational potential, that is, scientific outcomes, and how to improve, most importantly, animal welfare. But I suggest this information extends far beyond to the 3Rs themselves. First of all, replacement. By an improved understanding of the science of LED lighting technology and its species-specific application, we may replace inappropriate animal models with tissue culture and PDX models, which under the appropriate circumstances also respond to light at night, in vivo, and light-dark cycles, especially to LED light. Secondly, reduction. By improving research animal health and well-being through the use of LED lighting technology, we reduce the experimental repetition and also animal numbers. And finally, it's pretty obvious, LED lighting, in terms of its application and technologically important innovation, is clearly a refinement today. And I leave you with this thought. We know quite a bit about the impact of light at night on humans and animals. As I mentioned earlier, some of us in the lighting circadian fields have been investigating this for over 40 years. However, we really know very little about exposure to daytime LED light. There's so much to learn, especially how daytime LED light impacts the various species on the planet, as well as in the vivarium setting. I encourage everyone who has an opportunity and the wherewithal to take advantage of such studies in their own animals of interest. So for more information on this, I encourage folks to consider our 2019 comparative medicine paper and a 2023 and a 2024 JLAS review articles. I thank my great team at Tulane University, unfortunately approaching a hurricane, Francine, and a group at Thomas Jefferson University. Great collaboration over the many years, and for all the wonderful grants, especially the 2007 and 2018 GLASS awards that we were provided. Thank you, everybody. I provided my email address here at the base if you wish to ask me questions later.
USDA is an equal opportunity provider, employer, and lender.